This is what can happen to cells when placed in a hypertonic solution. Let's take a look at how we're able to achieve this. Now, first up, we're going to look at today just one particular kind of cells, and that's those cells found in eggs. Remember, eggs are organic. They're made of cells. Problem is they have this shell around the outside, so we're going to use some vinegar to get rid of that. We're going to need a cup where all this experiment's taking place, and then our hypertonic solution we are going to use is going to be corn syrup. Now, this is going to take us several days for osmosis to happen. Remember, osmosis is not something that happens instantly or extremely quickly. It takes time for all those molecules to move. So we're going to set up our experiment here. First up, by covering our egg in vinegar. Now, vinegar is going to act as a base, or an acid, to break down the calcium shell around that egg, leaving us a nice, soft, and permeable cell membrane to work with here. So we're going to take this, we're going to put some foil on the top to make sure that nothing escapes and it kind of smells, and we're going to let this sit for 24 hours and see what happens. The next day we come and check things out, and we have dissolved that eggshell and the vinegar has run its course here. We're going to dump off that vinegar and kind of rinse off our egg. And if you look at it, the egg is nice and squishy. It's retained some of that shell on it, just has a nice film. But we look inside and there is our cell membrane there. There you go, a rubber egg. We're going to place that rubber egg. Now remember, it's still raw on the inside, so it's not actually rubber. We're going to place that back into our cup, and then we're going to add our corn syrup to the mix. Now I'm going to add 50 milliliters of corn syrup here. Corn syrup is a very concentrated, it's a thick, very viscous substance, and so it should have a much higher concentration than the liquid that is found within the egg cell. So we're going to take those 50 milliliters, we're going to add them to our egg. You can see the viscosity here as I'm trying to pour that out. But once again, this is creating a hypertonic solution for our egg to rest in. And so think about what that means and which direction we would think molecules should go and make a prediction here. Based on what we know about equilibrium and diffusion and osmosis, which directions should those molecules move? And what should we see as a result after waiting another 24 hours? The next day, we're going to take a look at our egg, and we can already tell right here it is much more liquidy, more watery than the corn syrup was when we put things in. Now, the important part is we are going to gather how much liquid is left in the cup. So we're going to take our graduated cylinder. We are going to carefully remove the top of our cup, our container, and we're going to measure as much of that liquid as we can. It won't be perfect, as there'll be some that's stuck on the egg and stuck on the inside of the cup. But you can already tell that a lot more fluid has been drawn from the egg. Once we're done collecting our extracellular fluid here, we're left with our cell for the experiment, or the egg, and we can tell just by holding it here and squeezing it that it has lost a lot of fluid. So look at how pliable that thing is. It's squishy. It's still a raw egg in there, so be careful with that shell, but it has significantly deflated. It has lost a lot of that original its original fluid, and we can tell here by looking at it, it's got 83 milliliters of liquid came out of there. We put 50 in, so it pulled 33 milliliters of fluid out of the egg. And so we can compare our cell that's in hypertonic solution versus one that I had made on the side that was just left to be its regular cell. You can tell the size difference, that it has lost a significant amount of fluid in there. So there you have it. This is what happens when we put cells inside of a hypertonic solution, is that liquid is going to move into the surrounding area and out of the cell. If you like this experiment, Check out some more experiments listed below and subscribe to stay up to date on all kinds of different science lessons. Thanks for watching.